Okay, it's my pleasure to introduce our seminar speaker today, who is known to at least some of you. Those of you who are in the current EvoFunPath cohort will know Dr. Penelope Kahn through the course and other activities of the fellows. But yes, we're glad to have one of our current fellows, a postdoc fellow, presenting for us today. So Dr. Penelope Kahn is a current postdoctoral fellow in microbiology in Eliza Gerstein's lab here at the University of Manitoba. And Eliza is, unfortunately for us, at a conference, so I'm doing the introduction. I'm a fellow department member with Dr. Kahn. So having earned a PhD in zoology from the University of British Columbia very recently, I don't know, Penny, when did you defend? It's like December, right? Or
Yeah, November.
November, yeah. And prior to that, a bachelor's degree in biology from Macalester College in Minnesota, together with, interestingly, with minors in chemistry and anthropology. So a variety of study systems in the pre-PhD years. And I asked, what were some notable tidbits or something that I could share, and we'll have to ask for details later or in some other context. But one of the things that Penny mentioned was she had the unique experience of changing labs 2 years into her PhD program. So something to ask her about later. And the first chapter of her PhD dissertation was published in PNAS, which is, of course, a huge accomplishment. I'm going to just let you introduce your own title in case it's changed from the poster or whatever, but I'll turn it over to you and I look forward to your talk.
Great. Thank you. Okay. So everyone can see my slides, I guess. Cool. Yeah, so thank you so much for having me to give this seminar for the EvoFunPath program. I've really enjoyed my time so far in the program, and I've already learned so much. So I'm really pleased to be able to share a bit more about my background and the work I did during my PhD. So yeah, my title is Rapid Evolution of Saccharomyces cerevisiae in Severe and Complex Metal-Contaminated Environments. So not directly related to fungal pathogens, but I think there are going to be a lot of parallels that we can draw, and I'll point some of those out as we go. So as we know, a lot of environments on Earth are becoming kind of more extreme or experiencing additional and higher intensity of stressors because of anthropogenic impacts on the ecosystem. So humans have this kind of culture of like unmitigated consumption and transformation and redistribution of many of Earth's natural resources. And this can have detrimental effects to a lot of ecosystems. And importantly, we see that different combinations of stressors can have interactions that may increase their effects or potentially decrease them. And these interactions can be hard to predict and hard to understand what's going on. So this graph I'm showing, or this image I'm showing on the left, is from a paper that looked at human impacts on the deep sea ecosystems. And those lines in the middle um, are denoting synergies or interactions that increase the effects between different stressors that are being applied. And in my study system, I worked with heavy metals. There are a few reasons why. One of them is just that they are an ecologically relevant pollutant. Humans use large amounts of metals in many different applications, and we have to extract those out of the ground, which produces a lot of waste, as well as any waste products from whatever we use. And many times in metal contaminated environments, there's not just a single type of metal that is occurring at high levels, but there are often multiple different metals occurring at high levels in the same environment. Um, and using this kind of a panel of different metals allowed me to interrogate different stressors that have many similarities, but also differences between them. So yeah, again, like I was saying, it's not directly pathogen related, but if we think of the human body or any kind of host organism as like a complex ecosystem where there may be multiple selective pressures acting on the pathogen itself and different levels of different environmental factors in different areas of the body and so on. And we see rapid evolution happening in the context of the evolution of drug resistance, where there may be some population that when we apply a drug, kills most of it, but there may be some resistant individual or individuals that survive the drug and then replace that population with resistant individuals. And then we can also think about stressor interactions in the context of applying multiple drugs. There can be drug interactions that can increase the effect of um, the killing or whatever the goal of that drug is, or certain combinations can make those drugs be less effective. So kind of keeping this in mind when I'm talking about stressors in general, you can kind of potentially put that in the context of drugs and pathogens. So for a bit of background, I want to introduce you to this concept of evolutionary rescue, which is a rapid evolution scenario where there's some stable population that is well adapted to its environment. And when there's addition of some kind of environmental stress, the environment changes a lot and that population is no longer fit and the population size will crash and may go extinct unless there is the addition of some beneficial allele, either through mutation or migrating in from another population. And if that allele is present, then again, this population can be rescued, and the frequency of that adaptive allele will likely fix if the environment stays stable. So I'm just showing this concept again with a little fitness landscape cartoon. So we have phenotype space on the X and Y axis, and then on the Z axis is fitness. So we have this red population well adapted to its environment. If there's an environmental shift, a new combination of phenotypes is now fit in that environment, and our old population decreases in fitness. We expect that if there's a really large environmental shift and that peak moves very far away, that small effect mutations may not be enough to rescue that population before all of the individuals die and the population goes extinct. So we expect that if a population is going to survive this large environmental shift, large effect mutations, changes that affect the organism by a lot are going to be required. So if we have large effect mutations, it's more likely that that population will reach the new peak in a fewer number of steps. And this concept is relatively easy to think about if we're just considering a single stressor. And there's just one kind of ideal phenotype for that particular stressor. But what happens when there are multiple stressors in the same environment? How do they combine? Do we require two different mutations, one for each stressor, or is there some single mutation that could help with both? So these are the questions that I'm interested in. And I used this model system with yeast to study these questions. So Saccharomyces cerevisiae is the baker's yeast. It's a model eukaryote, which means there are extensive genomic resources, molecular resources. It's nice to work with for evolution experiments because it's freeze tolerant. So you can freeze down samples across the evolution experiment and then grow them all back up at the same time and experiment with them all together. My populations I worked with were haploid and reproduce asexually, and this occurs pretty fast, so we can see evolution happening in a matter of weeks. And since the genome is relatively small, bioinformatic analyses are relatively easy, which was really helpful for this large scale project. So in terms of how metals are used or seen by the cell, many metals are actually used in the cell in enzymes as cofactors. So an enzyme will need to bind a certain metal ion in order to gain the conformation it needs to act on its substrate. Um, and I have some examples of metal cofactors of the metals that I used in my experiments. So I use copper, cobalt, manganese, nickel, and zinc. I also use cadmium in my experiments, but that metal is not used in the cell, so it's not a cofactor for any enzymes. How metals can cause toxicity is when they reach high levels. So those metals that I referred to before, these are required in the cell. So the cell needs some mechanism of bringing them into the cell. But when there are too many metal ions around, they can become toxic. And some of the main mechanisms of toxicity that we think about for metal ions are oxidative stress. So metal ions will come into the cell and react with certain organic materials, produce reactive oxygen species, which can then go on to damage things like membranes, enzymes, and DNA. And then another important mechanism of toxicity is that when there's an excess of a certain metal ion, it can actually competitively exclude the native cofactor for an enzyme, kick it out, replace it. And in a lot of cases, binding of the wrong metal ion will not allow that enzyme to work on its substrate. So here are my 3 projects that I did in my thesis. The first was evolving yeast in the single metals and then seeing how those adaptations affect their cross-tolerance to other metals. And then I also did a double metal evolution experiment in my 3rd chapter. But before that, I had to figure out how those metals were interacting with each other to affect toxicity so that I could impose the right amount of selection pressure in the combination experiments. So the first project, evolution of cross-tolerance to metals in yeast. The question we're interested in, how do adaptations to one metal affect tolerance to others? This is the project that, as Matt said, was published in PNAS. And this was a really huge project that I collaborated with my collaborator, Dr. Anna Batsicalupo. She was a postdoc in the lab, in the auto lab when I was starting my PhD, when I was transferring into that lab. So we worked really closely together along with our really fantastic assistants, Yuli and Joel, to do this really massive experiment. Just a little bit of background, what I mean when I say cross-tolerance. We can also call this pleiotropy if you're familiar with that term. So we know that many genes affect multiple traits. So this is pleiotropy. A single gene can have effects on multiple traits. And if we take this hypothetical example of a non-selective transmembrane transporter that can transport both copper and zinc into the cell, if there's a mutation to that transporter, this could affect the growth in both copper stress and in zinc stress. And if that mutation affects those traits in the same way, so increasing the ability to grow in both copper and zinc, we're calling this cross-tolerance. And if it affects the associated traits in different ways, so for example, increasing growth in copper but decreasing growth in zinc, then this is cross-intolerance. And so this is a diagram of our experimental design. We were interested in looking at mutations, de novo mutations, so how evolutionary rescue can occur via de novo mutation. So we wanted to reduce the amount of genetic variation in our starting populations as much as possible. So we took a single colony isolate and propagated it in 240 wells, and then exposed those populations, which should be the same almost genetically, to these lethal doses of these 6 different metals. And every day we would go and visibly check the bottom of the wells in the plates to see if there was any growth. And most of the wells did not display any growth because those concentrations were so toxic, and either all the cells died or there were some dormant ones, but there was never a mutation that allowed for visible growth. But a small number of wells would experience, there would be some mutation that occurred that allowed for the cell that has a mutation to grow. And then right when we would see a visible amount of growth, we would sample it, freeze it down for use in our further experiments. So we got between, looks like with these numbers on the cells on the right, between 15 and 26 lines from each metal environment. And then we took all of those evolved lines and tested their growth rates in all of the different metals. And we also did whole genome sequencing to try to match mutations with fitness outcomes. So here's what the fitness data looks like. In this graph, I'm showing all of the growth rates of all of the evolved lines. So each tiny little gray dot is one evolved line. The colored circles are the average of the evolved lines that were evolved in a given metal environment. And then each column in the graph is the different test environments. So I have a black box around all of the lines tested in the ancestral environment that doesn't include excess metal. And the gray bubble is the ancestor of all of these evolved lines. So right away we can see that the evolved lines grow less well compared to the ancestor in the ancestral environment. So there's a trade-off occurring for these metal adaptations. So when there is not excess metal in the environment, the mutations that helped with excess metal are slowing the growth rate. One of the major patterns we saw in this fitness data were that certain environments selected for mutations that were kind of generally helpful for lots of different metals, and others selected for mutations that were kind of more specific to the metal that they were experiencing. So if we look at the cobalt lines, which are in blue, we can see that they were able to grow in almost all of these metals and did so at a comparable rate to any of the others. And we can compare that to the manganese lines in pink, which were able to grow quite well in the manganese environment, but did not grow well in any of the other environments. So we'll kind of dig into this in a little bit when we get to the genetics. First, I want to point out that we tried to actually, before doing the experiment, we wanted to try to predict the cross tolerance we would see by looking at different ways that the metals are similar. So we thought that if 2 metals are more similar to each other, there would be a greater chance that cross tolerance would evolve. And so we collected data based on 3 kind of metrics of metal similarity that we came up with. So the first was the oxidation reduction potential. So this is how reactive each metal is, and we actually measured with a probe the oxidative reductive potential of the metal media that we used. The second piece of information was the known resistance genes on the Saccharomyces genome database. So there is all this information from single gene knockout studies where they'll knock out every single gene and then grow these mutants in different environments, including different metal environments. And they can see the effects of different mutations on the phenotype of growing in metal. So the overlap in the effects of these different genes was our second kind of similarity metric. And then the third one we looked at was the co-occurrence of different metals in natural environments. So we took a bunch of the data from the US Geological Survey and found the correlation of each pair of metal in across, I think it was 77,000 different sample sites, um, kind of with the idea that if two metals are co-occurring in the same environment more often, organisms in general may have common mechanisms for resistance between those two. So I'll tell you that none of these similarity metrics were able to significantly predict the cross tolerance. So we weren't able to predict cross tolerance, which I think just goes to point to the stochastic nature of evolution. It's very hard to predict. Getting into the genetics, so the way that our experiment was designed, we wanted to minimize the number of mutations that the lines were accumulating before we sampled them. So we sampled them right when we saw the growth so that there wouldn't be all these mutations accumulating because we were interested in being able to match a mutation to a phenotype and especially in that kind of first step of evolutionary rescue. So these mutations may not be the most fit mutations, but they're just a first step that allowed those populations to be rescued. And we saw that our efforts to minimize the number of mutations was overall pretty successful. They mostly had between 1 and 8 mutations or so. The average number of mutations per evolved line was 3. And we did see that cobalt lines tended to have a higher number of mutations than lines from other metals. We also saw a few examples of hypermutator lines from the manganese environment that had mutations in DNA repair genes, and they accumulated many. Oh, and I should have said these are genic mutations, so mutations in open reading frames that we're looking at right now. When I did the analysis of the functions of those genes that were mutated, so I looked at the gene ontology terms and constructed this kind of hierarchical map of the significantly overrepresented gene ontology terms in our mutation set. So in terms of like the function of these genes, a lot of them had to do with kinase activity, transferase activity. So this is like in general kind of a lot to do with probably cell communication and regulation of cellular processes. We also saw significant overrepresentation of, these MAP kinase, so kind of stress response processes. And then, in cellular process, we have these kind of large categories. So biological regulation, regulation of cell processes. So this really toxic environment required, in a lot of cases, kind of like whole cell regulation changes, which makes sense. We also saw that the vacuole was significantly important, and particularly this vacuole or transport chaperone complex, which I will touch on a bit more in the slide after this. And so I wanted to show that this map is agnostic to the effect of the mutations. This is including all mutations. So I wanted to know kind of the distribution of effects that these mutations were having within each Go term. So here I'm showing all of the significantly overrepresented Go terms, and green are silent mutations, yellow are missense, and then red are these really large effect mutations that may cause loss of function. And we see that this polyphosphate biosynthetic process and this vacuolar transport chaperone complex, which are related to each other, have a lot of loss of function. And so I'm going to paint a little picture for you about why this phosphate metabolism might be important. So we found that our lines that had the broadest cross tolerance, so we're able to handle many different metals, we kept seeing this phosphate metabolism come up. And so this is my interpretation based on all of the reading I did, that it's really about this Pho84 transporter. So this is a high affinity phosphate transporter, Pho84, but it also is capable of transporting these divalent metal ions. And so we have this vacuolar transport complex that transforms phosphate monomers into polyphosphate in the vacuole. And then we also saw this inositol phosphatase gene was mutated often. So the the kind of changes that occur when these have a loss of function go towards removing this Pho84 transporter. So when we lose the vacuolar transport complex, we're not transforming phosphate monomers into polyphosphate. We have this buildup of phosphate monomers in the cell. And then we get this signaling to stop producing Pho84 and actually to degrade this Pho84 transporter. So these metal ions can no longer enter through that transporter. So I initially was expecting that most of these metal transport or most of the metal adaptive mutations would have something specifically to do with um, genes that showed up in like a metal context, like that were directly um, kind of interacting with metals. But in fact, we found a lot of other processes that are tangentially related to metal metabolism, but not directly. So it kind of, again, goes to show the complexity of the cell. It's kind of hard to predict exactly what's going to happen. And I'm going to leave conclusions to the end just for time's sake. So getting into the second project, again, I was quantifying the effect of these metal interactions on the toxicity of the environment. So this is up on bioRxiv already. I'm almost done finishing the manuscript to submit. Sally, my supervisor for my PhD, she helps me a lot with the math that you'll see in this part. I'm not necessarily a math person, but I got some good experience with it. And then again, my undergrad assistants were really helpful for the lab work here. So initially I just wanted to go straight from single metal experiments into double metal experiments. And I took the concentrations that I used in my single metal experiments, like decreased the toxicity by what I thought was half and put them together. And in all of my pilots, I was not seeing evolutionary rescue where we only get a few wells showing growth. I kept seeing either everything in the plate was growing or nothing in the plate was growing. So I decided I needed to dive in deeper to what's going on when I'm combining these metals so that I could choose concentrations that would result in this evolutionary rescue pattern. So the way that I did this was to measure the growth rates of the same the same ancestral line used in my first experiment in comparing their growth in two individual metals and then in their combination at the same concentrations. And I did so at a gradient of concentrations to try to see this interaction across different stressor intensities. And I wanted at each concentration level for there to be approximately equal stress level from each metal. And so I did this for all of the 15 combinations of the 6 metals I used in my first experiment. So I'm going to show this graph on the next slide with all 15 concentrations. So that's what this looks like. And we can see a lot of differences between different combinations. So some combinations did not allow for growth at any concentration level, even when the single metals by themselves are not very toxic and the growth rate, which is on the y-axis, is pretty high. When they're put together, there's no growth at all. Some combinations allow for equal amounts of growth when both are in the same environment, or even better growth when there's two metals there. And then the rest are kind of somewhere in between. So I wanted to be able to quantify this, what these patterns that we're seeing here. So to do that, we had to come up with a null expectation. Or I guess when people are doing mixture toxicity analyses, they compare different combinations to a null model. And the one that we used was concentration addition, which assumes that if two different metals were actually the same metal, their combined effect should be equivalent to doubling that effective concentration of either one. And so we have this deviation function with this A term that describes the amount of deviation from this null expectation, where a negative A is pointing out synergistic toxicity, where the combination is more toxic than expected, and a positive A is antagonistic toxicity, where the combination is less toxic than expected. And then an A of 0 would be this additive expectation. So we did this analysis for all of the 15 combinations. I'm showing 6 examples of this kind of 3D representation of those growth rates that I showed in that kind of grid. And the A value is given above each of the graphs. So we can see with the negative A's, the synergistic combinations, there's this kind of concave shape where the growth when there are two separate metals is lower than when they're by themselves. The ones in the middle, cadmium cobalt and cadmium zinc, the A value is not significantly different from 0. So this is kind of an additive expectation, and we can see the graph kind of drops off evenly across the whole thing. And then on the right are 2 examples of antagonistic combinations where we see this convex shape where there's kind of a higher growth of the in the combined metals compared to the single ones. And then below that, I'm just showing the range of A values in all of these combinations that we tested. So if we look at that data a little bit differently, we can pull out patterns of the effects of individual metals on the combination. So I'm showing the A value on the y-axis here, and each single metal on the x-axis. So for each column, we're looking at a single metal. For example, the left column is manganese, and then all of the shapes in that column are metals they're combined with. And then wherever they are is the A value. And we can see that, for example, all of the combinations with manganese have a positive A value. The white circle is the average of all of the different combinations, the average A value. The black dotted line or dashed line is the average of the combinations with cadmium, cobalt, and zinc, which are kind of the neutral metals. So going through this, we can see that all of the combinations with manganese are antagonistic. Almost all the combinations with copper are synergistic, except for the one with manganese. And then the nickel combinations are mostly antagonistic, except the one with copper. The ones with, for zinc, cadmium, and cobalt, the A value of the combination depends on which metal they're paired with. So for all of these metals, the manganese and nickel combinations are antagonistic, but the combination with copper is synergistic. And when they're paired with each other, the A value is 0. So they're kind of not strong drivers of this interaction. They're kind of neutral and combine additively with each other. So by looking at the dashed line, we can see that there's like a different level of strength of these interactions between the metals, where manganese would be like a really strong antagonistic driver, copper would be a synergistic driver, and then nickel would be a weaker antagonistic driver. I guess I'll give some potential reasons for this. So in my reading, I found that manganese, I think that manganese is antagonistic, is decreasing the toxicity of the environment because manganese is able to form these small molecule complexes that can scavenge reactive oxygen species. So they're kind of removing one of the major stressors in the environment, imposed by the other metal that's there. Copper, I found there's evidence that it permeabilizes the membrane, which may be allowing more metal ions to enter the cell, which is my idea of why it's synergistic and making the environment more toxic because just more metals are able to enter. So those are kind of speculative based on reading, but some possible explanations. So this A value is not super or it's kind of, it takes a lot of data and computational effort to calculate. So I was interested in coming up with a new metric that was a little bit more intuitive and required much less computation, and much less sampling at different concentrations and so on. So we have this metric that we devised called delta, or cumulative toxicity, and it compares the growth rate in the difference between the growth rate in the combinations and the single metals divided by, so depending on how toxic the single metals are. So it's able to be applied across different stressor intensities as well. So a delta of 1 would mean there's no growth in the combination. There's like a maximum amount of difference between the single metals and the double metal. A delta of 0 means there's no difference between the single metals and the double metal. And we can see that in the graph on the right, I'm showing the relationship between that A value that is like a commonly accepted term, and our new delta value, and show this significant relationship, which kind of validates that this metric is saying something true about these combinations, and may potentially be more useful for ecological applications. So if people want to know if 2 pesticides, for example, were worse or better than just one, then they could use this metric potentially. And then one other interesting thing I want to point out is that those metal similarity metrics that I talked about in the first chapter that weren't able to predict the cross tolerance that evolved, we actually do see significant relationships between the A value, so the interaction, and the redox potential and the known resistance genes. So more synergistic combinations tend to have a greater difference in the oxidative reductive potential and also less overlap in those resistance genes. So that's interesting. Okay, going to project 3, doing evolutionary rescue in these combinations. How do those different interactions affect evolutionary outcomes? Let's see how much time I have. Can't see my. Okay, great. So this is the least finished project. I still have to do the genomics on these evolved lines. So it's kind of half of a story right now, but I think that it's still interesting enough to show. So these were my undergrad assistants for this project, Jordan and Keon. Thank you to them. Yeah, so here again, when there are 2 different stressors in the same environment, they may impose overlapping selective pressures or completely different selective pressures. So how do organisms evolve and adapt in these scenarios? So the the experimental design was similar to the first experiment. Since we had more environments, I had fewer populations. So I did 80 populations, again, all from that same ancestor that I used in the first experiment, and then expose them to lethal combinations of metal. And then again, checked the plate every day to see if there was new growth. And I'll say that choosing the concentrations for this experiment was pretty challenging. I had two criteria I wanted to achieve for these concentrations. First, I wanted the concentrations of each single metal to produce equivalent stress on their own. So to impose a similar growth rate just in the single case. And then when combined, I wanted the stress level to be just past this tipping point between being able to support growth or not being able to support growth, so that I would see this evolutionary rescue pattern in the plates. So choosing those concentrations, again, was difficult. I don't have time to talk about exactly how I did that, but I will say that just because we didn't end up getting evolutionary rescue in all 15 combinations. So I got successful numbers of wells with growth in 8 of the 15 combinations, which are shown here in these graphs. So we got evolutionary rescue in 2 of the 4 synergistic combinations, 6 of the 8 antagonistic combinations, and 0 of the additive combinations. And then on the right, I'm showing the numbers of petites versus grands. So yeast, Saccharomyces cerevisiae, is a facultative. It prefers to ferment sugars, but once it's used up all the sugars, it switches to respiration. So it can both ferment and respire. But it doesn't need to be able to ferment. So if it loses, or it doesn't need to be able to respire. So if it loses function of the mitochondria, it can still ferment sugars and get energy. So we often see in evolution experiments with yeast the spontaneous production of this petite phenotype, which doesn't have mitochondrial function and cannot respire. And we saw that was pretty common in a lot of the combinations, but some of them it was less common. So I tested the growth rates of all of these evolved lines in 4 environments. So each of the single metals on their own, the combination environment, and then the ancestral environment. So again, the single metals are on the left and right side of the graph. The growth rate in the combination environment, so the evolution environment and the ancestral environment are both in the middle. The evolution environment with a combination of metals is the closed circle, and the growth rate in the ancestral environment, or YPAD, is the open circle. And I'm going to show 2 versions of this. So one will have max growth rates R, and then I'm also transforming the data so that we are looking at growth rate relative to the ancestor. So the 0 line in these relative fitness graphs represents the growth rates of the ancestor in all of these environments. And then if the evolved line has a higher growth rate, it's growing faster than the ancestor. If it's below, it's growing slower than the ancestor. Okay. So that's what this data looks like for all of those 8 combinations. And there is a lot going on here. I have these graphs arranged from left to right with synergism to antagonism, shown in panel C here with the A values. And we see that in general, there are a few patterns here. So one is that as we go from left to right, the growth in the single metals, so on the outside of the graph, seems to be increasing in this relative fitness plot. And then we also see kind of that some of the lines have an asymmetric shape for growth in each of the single metals. So many of them are kind of gaining adaptations that help more with one metal than with another. So I'm gonna dig into this a little bit. So here I'm showing graphs where we have A on the x-axis and then the fitness in each of the 4 test environments on the y-axis. And so for panels A and B, that top level, this is the growth in the combination environment, which I'm calling the benefit of the adaptation. And then on the right is the growth in the ancestral environment, which is the cost of the adaptation. So the type of interaction that's occurring is not different between, or does not cause a difference in the benefit or cost of these mutations. But we do see that there is a significant relationship between A, the interaction value, and growth in the individual metals. So combinations that have more antagonistic interactions allow for adaptation to each of the single metals, whereas more synergistic ones, the adaptation is more specific to the combination environment rather than gaining fitness in the single metals. And remember that there are different interaction strengths here between the different metals. And since we didn't have rescue in any of the additive combinations, each of the combinations has like a weaker and a stronger interactor involved. So here I'm showing growth or relative fitness in the stronger interactor on the X and relative fitness in the weaker interactor on the Y. And we can see that in general, many of these lines have a higher gain in fitness for that stronger interactor, which in many cases is manganese or copper. And on the right, so we see this strong pattern that the stronger interactor there's a greater gain in fitness for that metal. But on the right, I'm showing that there is not a significant relationship between the magnitude of that interaction and how big of a difference in fitness there is. So like how asymmetric that graph is. But we do have a significant intercept. So the positive intercept implies that there is this significant pattern of having better growth in the stronger interactor, but it doesn't depend on the type of interaction. So to draw some overarching conclusions, there are a huge diversity of genetic mechanisms and fitness consequences of evolutionary rescue. We had a huge amount of replication and saw a huge amount of strategies that came with that. But in most cases, those adaptive mutations for metal came with trade-offs for the ancestral environment. We found that adaptation was really hard to predict and cross-tolerance was hard to predict. And it occurred through these kind of unexpected pathways in a lot of cases. But we did see that those metal similarity metrics predicted stressor interaction, so that may be more straightforward. In terms of why didn't we get rescue in any of the additive combinations? One of the kind of speculative ideas I have for that is that interactions, even synergistic interactions, may actually facilitate adaptation compared to when there's no interaction by kind of transforming and consolidating selection pressures, which may require fewer mutations. So maybe in those additive combinations, you need mutations for each stressor, but when there's an interaction, maybe it reduces the number of mutations you need. And then, yeah, very soon, hopefully we'll be able to get genomic data for the double metal experiments and be able to compare that to the single metal experiments. I think that will be very informative for this picture as a whole. So I want to thank my supervisor and my collaborators, my committee, my lab at UBC, and all my funding agencies. And I would be happy to take a few questions.
Thank you, Penny. Very large data sets there. Impressive amount of work.
Thank you. Yeah, yeah, it was a lot.
I don't know who we have who's not a fellow. Maybe we'll ask. Yeah, we've got at least
A few
Milton
hands
joining
raised.
us from. Yeah.
Yeah, I think Milton was first, and then Christian.
Yeah, thanks. Really nice work. I was wondering if you had looked at all at metal chelation. Does metal chelation stop these interactions? Do you see any mutations in chelating genes?
Yeah, that's a really good question. It's a whole part of the story from the first chapter that I didn't have time to talk about today. But yeah, so one of the metal chelators, this CUP1 gene, which is the metal metallothionein, it can bind copper. There's also been evidence that it can bind cadmium, but it's kind of mixed how effective that is. And we wanted to look, we had evidence from a previous paper that showed that copy number of this CUP1 was increasing over like in exposure and evolution in copper at least. Um, and that increased copy numbers was helpful for higher levels of metal. But in our data, we didn't find that. So we did see a big variation in copy number of the CUP1 gene, but it wasn't associated, like it didn't have a linear relationship with tolerance to copper or any of the other metals. Um, so it was kind of a confusing story a bit with that. But yeah, we did look at it. Um, yeah, there was so much data that I could have, this project could have been really never ending, and I could have like dug in deeper to like the glutathione response and all of that. But yeah, we didn't end up finding like a strong relationship between that, but we did look at it. Yeah, it's a good question. I'm not
Interesting. I
sure.
wonder if
Oh,
you
go
had
ahead.
done the transcriptome, because if it's a copy number variant, you might not see that with isogenic lines kind of going in. Might not be enough time to get a duplication, but you might see a transcriptomic shift. So if you didn't do transcriptomics and you eliminated intergenic variants, you might not see that. So anyway, lots and lots to do. Very exciting. Thank you.
Yeah, thank you. Yeah, we didn't do transcriptomics. That would provide a lot clearer of a picture of yeah what's like going on in the cell. But with the scale of the data, we yeah just had time to do genic changes. Thanks. I think Christian. Yeah.
Yes. Awesome talk. This was really, really interesting. I had a quick question for when you were doing the evaluation on your tolerance for, I guess I'm doing a similar project, and I was curious if one of your concerns when you were making the media compositions for stressing the Saccharomyces was pH. Um, did you pH normalize during your experiments? I was just curious.
We didn't. Yeah, so there it's a good question. For example, copper tends to impose a much lower pH than the other
Yeah.
metals. Um, but we decided that the pH differences were a feature of that
Hmm.
metal. So we decided not to. We did play around
Okay.
with the idea of normalizing pH to remove that as a factor, but
Gotcha.
decided to lump it in with all of the other stressors imposed.
Okay, and another question, if I can, have you thought of like evaluating any of the strains that have like a differential growth on act solid media to see if there's a physical morphology that you can observe other than just OD and growth rate?
Yeah, that's a good question. We didn't end up like growing them on we grew them on solid media to test for petites, like the
Hmm.
respiratory phenotype. But we didn't do like colony phenotype.
Mm.
I did take images of them like with light microscopy
Oh,
and
okay.
tried to do like a cell size
Hmm.
analysis, but didn't end up finding any significant patterns with like cell size. There were some lines that I noticed had more like of
Clumpy?
a flocculent phenotype.
Yeah.
Yeah, so they were more clumpy. Also ones that seemed like like there were kind of different flocculant phenotypes. So some
Hmm.
that looked like they were staying together and some that looked like they were sticking together. So like not breaking off after division. Uh,
Yeah.
and then some were, but I didn't have time to
I
go.
get it. Yeah. This is already a lot of work. Um, and I guess, yeah, thank you.
Yeah, thank you.
So we are at the top of the hour here. We should let any non-fellows who have other commitments go and fulfill those commitments. But really appreciated the talk, Penny. Excellent work, and thank you. And I'm going to sign off. I don't know. Okay, here comes Marie Helen with some instructions, maybe.
Well, no, it's just that I'll stop the recording, and Penelope, if you want to take maybe 2-3 minutes, and then we'll come back with the fellows.
Yeah, that'll be great. I'll be back at 11:05.
Okay, perfect.
Okay.
Thank you, everyone.
Yeah.